Welcome to our exploration of the cell cycle and its regulation, a fundamental process in biology that ensures the proper growth and division of cells. The cell cycle, first described by Howard and Pele in 1953, is a series of stages that a cell goes through from one division to the next. During this cycle, the cell grows and prepares for division, alternating between genome doubling in the synthesis phase and genome halving during mitosis. The cell cycle is completed in two main phases, the interphase, which includes G1, S, and G2 phases, and the M phase, which involves nuclear and cytoplasmic division. This process leads to the doubling and halving of the genome, essential for cell division. This diagram illustrates the different phases of the cell cycle, highlighting the growth and division processes that occur as the cell progresses through G1, S, G2, and M phases. In the G1 phase, the cell synthesizes enzymes for DNA replication, RNA for transcription and translation, and raw materials for DNA duplication. This phase is crucial for cell growth and preparation for the next phase. During the S phase, DNA replication occurs, centrioles divide in animal cells, and histone proteins are synthesized, preparing the cell for the subsequent G2 phase. The G2 phase involves the synthesis of proteins for spindle formation, plasma membrane formation, and the doubling of cell organelles. ATP synthesis increases to support these activities, preparing the cell for mitosis. The M phase includes karyokinesis, where the nucleus divides through prophase, metaphase, anaphase, and telophase, and cytokinesis, where the cytoplasm divides, forming two daughter cells. This visual representation shows the stages of the cell cycle, from interphase through cytokinesis, illustrating the progression and division of cells. After cell division, daughter cells begin a new cycle with interphase. Some cells may enter a quiescent phase, pausing the cycle, but can re-enter when needed, such as during tissue formation. This graph depicts the changes in DNA content during the cell cycle phases, highlighting the doubling and halving of DNA as the cell progresses through G1, S, G2, and M phases. The cell cycle is tightly regulated by molecules that ensure proper progression and prevent uncontrolled division. Cyclins and cyclin-dependent kinases are key regulators, with their discovery earning a Nobel Prize in 2001. Cyclins and cyclin-dependent kinases work together to control the cell cycle. Their levels fluctuate with the cycle stages, ensuring precise regulation of cell division processes. This table outlines the Cyclin CDK complexes formed during cell cycle regulation, detailing their functions in preparing the cell for division and activating DNA replication and mitosis. Cell cycle checkpoints monitor and regulate cycle progress, ensuring DNA repair before the cell proceeds to the next phase. The G1 checkpoint allows for DNA damage repair before S phase entry. The S phase checkpoint ensures DNA integrity, promoting repair of replication errors. This quality control is crucial before the cell moves to the next phase. The G2 checkpoint prevents cells with unreplicated DNA from entering mitosis, ensuring complete genome replication. The spindle assembly checkpoint ensures accurate chromosome distribution during mitosis. Checkpoints rely on proteins like p53, which senses DNA damage and halts the cycle for repair. If damage is irreparable, the cell undergoes apoptosis, preventing faulty cell division. Thank you for joining us in exploring the intricate processes of the cell cycle and its regulation, a vital aspect of cellular biology.